I'm very happy to start our third MMP seminar, and my name is Seirin from Kyoto University and one of organizer of this MMP seminar. And then the other organizer is the Dr. So Miyoshi is there. Okay.
Hi, good morning, good afternoon, and good evening, everyone. I'm Somios from Pfizer R&D Japan. So I'm co-organizing this seminar with Professor Senrei. Thank you very much.
Thank you. And then before the seminar, I would like to introduce our really nice data advisors. We have two advisors from Pfizer and Society of International Society of Pharmacology, and then three advisors from Kyoto University. Okay, so first let's start with CJ.
Hi, everyone. Thanks, Sarah, and thanks, So, for the introduction to be here and part of the advisory committee. And I'm very excited to be here tonight to hear Professor Zhao's talk, one of his biggest fans, and really excited for what we're going to hear tonight.
Thank you. And then Dr. Kawai from Pfizer.
Yes, hello, I'm Nori Kawai, Clinical Research in Pfizer R&D Japan. So actually, I'm very interested in the application of the QSP modeling to translation research and clinical development, especially for new modalities. So I'm very looking forward to this seminar. Thank you very much.
Thank you. And then I would like to introduce our advisor from Kyoto University, Professor Motoko Yanagida.
Hi, it's Motoko Yanagida from Kyoto University, Japan. I'm a nephrologist, and I'm an outsider of math, but I'm quite interested in the combination of math and medicine. So I'm looking forward to the seminar of Dr. Zhao.
Thank you. And then Professor Kenji Kawashima.
Nice to see you all. This is Kenji Kawashima from Kyoto University. I'm also very excited to join this seminar. Thank you.
Thank you very much. And then actually we have one more advisor from Kyoto University, is Professor Kawamoto, but unfortunately he couldn't join in this time, but he will join the next seminar. And then this is a really exciting moment. Dr. So, please introduce our super nice speaker, Professor Chen Zhao.
Yes. Please let me introduce Professor Chen Zhao from Nanjing Medical University. Professor Chen Zhao obtained his bachelor degree in biomedical engineering from Washington University in St. Louis, and his PhD in biomedical engineering from Johns Hopkins School of Medicine. His major area of interest is QSP and mechanistic modeling in biomedical research and drug development. And he has authored over 30 publications in recent years and serves as the PI for more than 20 academic industry grants. So Professor Chen Zhao is also authoring team member for several recent model-related drug development guidance document issued by China regulatory agency, CDE, and he's a leading advocate of QSP, not only in China, but also globally. So he also has received numerous research awards, including the AHA Predoctoral Fellowship and Johns Hopkins Medicine Annual Young Investigator Award, and Jangus May 4th Medal, and Jangus Distinguished Professor in Biomedicine. He also served editorial position for international journals such as Current Opinion in Pharmacology, for Frontiers in Systems Biology, and Current Research in Pharmacology and Drug Discovery. So we are very great honored to have Professor Chen Zhao here. Today's his presentation title is his title is Mechanistic QSP Modeling for Translational Drug Target Research and Clinical Development of New Therapeutic modalities. Professor Chen, please.
Thank you all for the very kind introduction, and it's my great pleasure to join this seminar and present some of our recent works. So let me just share my screen. Okay. Okay, I think it's working, right?
Yeah, it's
Okay.
working. Yeah.
So, okay, for the next 45 minutes or so, I'm going to go through some of our recent work in this area. So as you know, I'm Chen Zhao from Nanjing Medical University, and this is my disclaimer. So I see that many of the audience may not be drug developers, so I figure it'd be good to give a very quick introduction of QSP, what it is. So as you know, ISOP is basically leading the effort of model-informed drug development. So the drug development field has been moving or has been improving, but one thing that has not been really improving is the drug success rates during the clinical development stages. So according to some numbers, so the clinical success rates from phase 1 all the way to marketing or regulatory approval has remained low or around 10% or 10, 13%-ish over the past 10, 15 years. This is from a bio survey, so from the organization Bio. This is from another 2018 paper where the authors categorized the drug development efforts into specific disease areas, and you see that the numbers vary, but the overall rates are more or less the same, around 10%. So the concept of QSP arose around 2000-ish, but has been formally published during in this first time in this 2011 NIH white paper, where a group of university professors in the US and together with industry professionals as well as regulatory representatives, these three parties, they came together and wrote up this very landmark paper of QSP. It's like 50-page-ish, but it's very comprehensive, including the information you need to know about what it is and why we need it, and how to educate people that can master it. But the core concept, of course, is to use the latest methodology, which is systems biology modeling, to study concepts in pharmacology, which can be attributed to basically PK and PD, the pharmacokinetics and pharmacodynamics. But the merger of the two form this new discipline of QSP, quantitative system pharmacology, that are believed to play critical roles in the drug development in nowadays, and it has proven so. So for the application scenarios, as this area is meant for drug development, but also for drug research, as lately people have discovered that model informed can also play a role in early stage drug research. So for the clinical side, when we advance a drug into the clinical trials, we need to decide its dose, its appropriate dosing regimen, what combination should we use, and what are the target patient population. So by simulating the disease at the clinical level, we can prospectively basically estimate or assess the potential efficacy of this drug or any drug combination in this target population, so we can reversely decide the appropriate dose, appropriate regimen, appropriate combination, and also the potential biomarkers that may be helpful in stratifying the patients. But of course, when we have this prediction in mind preclinically, we can already give very much information to the preclinical development of any drugs, basically selecting a target, if the target is the right in this network, selecting the right drug, or optimizing the affinity of the proposed drug. Is this antibody having suitable affinity, or is this bispecific having suitable affinity on this hand and on that hand? Things like that. So recently, the MIDD concept has more or less evolved into the MIDDD. Basically, you have the discovery part, and also people are thinking of adding the decision-making part into the MIDD paradigm. So over the years, there has been many practice. Many have proven important in the clinical development of blockbuster drugs. And there are also several, or at least, I think, maybe 10-ish industry surveys regarding the application of QSP from the industry side, from the FDA side, or from both. I just listed 5 over here, where you can see these 5 are more or less 1, 2, 3, 4 from the industry side. The third one is from the FDA side, but they surveyed different aspects of the QSP field, from either internal use or communication with the FDA regarding the clinical submissions or clinical evaluations, or from what softwares does it use? What's the difference between this and traditional PKPD, or in specific areas such as immuno-oncology? And this is the latest FDA summary of QSP from their perspective of, reviewing. As you can see, there hasn't been too much submissions early on, but later in 2020, by the end of 2023, so there has been over 80 submissions to the FDA where they think this is important. And also for specific areas such as rare diseases, the number is also rising. And we also see establishment of QSP teams, originally only in the big pharma, but now more and more and emerging in the biotechs and the new CROs. So another aspect of importance of QSP in the drug development area is the latest roadmaps proposed by the FDA. For example, in the 2024, there is this new Quantitative Medicine Center of Excellence formed within the FDA, where they very much focus on mechanistic modeling and clinical trial simulations, which are closely related to QSP. And last year, we saw this new plan of phasing out animal testing, or gradually phasing out animal testing, where the FDA have listed 3 major areas of the new alternative methods, the NAMs. And among them, one big category is the in silico tools. And among the in silico tools, also you see the QSP being listed as one of the 4 approaches. So, for the next section, I'm going to go over some of our recent work for the QSP, both on the drug discovery side and also on the clinical development side. But for the clinical development side, I'm going to focus more on the advanced therapies, more or less the ATMPs, or cell and gene therapies. So my group has been focusing on a wide range of disease areas. But for today, I'm going to go over some examples in the vascular and CNS, and then oncology and inflammatory diseases, where I'm going to start with cell therapies and then briefly on vaccines, or both prophylactic and therapeutic. So my personal journey of research starts with basically cardiovascular area. So I joined Hopkins in 2014. From then on, I started this research topic in peripheral arterial disease, where it is a very common disease, but unfortunately, it has no drugs specific for the cause of the disease. The current standard of care is basically antiplatelet. So you don't want extra cardiovascular occlusions due to the peripheral arterial disease, but there's no drugs that can target the cause of the disease, which is limb ischemia. So over the time, some of the patients with PAD will progress into the end stage, which is called critical limb ischemia, and a portion of those would require, unfortunately, amputation, and they have poor prognosis, even in terms of life expectancy. The original idea of this project back when I was at Hopkins is to develop a computational model, or more or less the QSP type model, where we model the pathophysiology of PAD, and with the hope that we can maybe develop some new drug targets for PAD. So with that, we focus on, I chose the simple path, which is to focus on the mouse model of PAD. But even for mouse models, it actually is a very complex experimental procedure that requires usually up to a month because you have to ligate the femoral artery of the mouse, and then you wait for a quite long period and you observe the perfusion recovery of the mouse. So that is the most commonly used animal model for PED for drug target discovery and also. So we chose to computationally model the experimental model for PAD with the hope that we can do actually quick experiments for model validations, for computational model validations. So we established this multi-skill model that included both signaling pathways of the essential cells that play a role in the basically in the tissue environment of this skeletal muscle. And also we included the key cytokines and the endothelial cells, the myocytes, the macrophages, how they interplay with each other, and how they lead to the final observation endpoint, which is the perfusion recovery or the extent of angiogenesis in this skeletal muscle. So we combined all of them into a big model. And we use multi-skill data for the model calibration because the goal is to identify some new targets. So we first basically pick all the information that we can have from the literature and use them to calibrate the model. That included both signal transduction, cell dynamics during this time course of the ligation recovery, and also some endpoint markers such as the how much perfusion do we have at the mouse level, tissue level, the biomarkers for necrosis, the oxygen level, protein expression, etc. And after that, so we did some analysis first, only for the macrophage polarizations, just for the research interests. But later on, we went on to see if we can really identify some new targets. So we first did an sensitivity analysis, looking into what are the essential nodes that can perturb this perfusion endpoint. So we identified a bunch of them, but this field has been advanced. This field has been studied, but without very much success. But unfortunately, there are many of the targets being already tested in animals. So the only thing that wasn't tested but came up in our model prediction is this necrosis or necroptosis pathway that was predicted to significantly enhance perfusion recovery. But no one has done experiments on this. So we went on to do some experiments. We selected this established NEC1S, so a inhibitor for the necroptosis pathway. So we added this drug into the mouse, and we have a control group as well, the DMSO control group. And after that, so we did observe that this drug, so if you compare this small leg to this small leg, we did see that it enhances the perfusion recovery at the early stage to a very much significant extent. So this at least proves that the drug target identified by our target is valid in terms of the translational efficacy. And we also seek to determine if we can, in some way, validate its potential in the clinical samples. So we did collect some clinical samples with the help of clinicians. So by comparing the gene expression in the patients that have advanced to the endpoints and stages of the PAD, which are the critical limb ischemia, to the patients that are normal, the healthy adults, we see that the pathways that are closely associated with the necroptosis, basically the RIPK and MLKL, are highly overexpressed in the critical limb ischemia patients. And also, if we just do gene expression in the mouse, it also shows that this is highly upregulated. So at least this has some relevancy in the clinical stage. So this is part of the. So this the results that I just showed results. So in this publication in 2022. But unfortunately, we haven't advanced it to the clinical stage. For example, we haven't tried to really model the PAD patients at the clinical level. Also, nor to looking at the clinical endpoints such as the 6-minute walking test or things like that, which I think is the pity, due to I basically moved later back to China. But this at least serve as a good paradigm for model-informed translational research in terms of drug target development. So later on, after I went back to China, so I started my own group. we are now very much into the CNS area, but also the vascular side, the ischemic stroke, which is much more hard to treat in terms of neuroprotection. And also there aren't many drugs approved for that, which basically is only 1 or 2 drugs approved. So ischemic stroke, many of you should be quite familiar with that. It starts with the occlusion of your arteries, and then your brain partially dead. And then you have to basically recanalize this occlude artery by some drugs, TPA or surgery. But over the time, that doesn't really get you back to the normal state. Or most of the patients, they don't restore 100% to the normal state, because this very short term, or maybe a few hours, maybe 20 hours-ish, ischemic status of your brain can lead to really unexpected damage, because it leads to various stages of events that involves cell interplays of neurons, of endothelial cells, which are the microvascular brain endothelial cells, also the microglia, the astrocytes, also the oligodendrocytes. So very complex cellular interplays during this acute and chronic stages that involves different aspects of inflammation, neuronal death, and oxidative stress, etc. So for those that are not very familiar with the diagnosis or the evaluation of stroke, usually in patients and in mouse, you would have a ischemic or infarct core at the very beginning. So the area is 100% dead. So basically, it was determined from the very beginning. But you have a surrounding region called penumbra that is not really, this still can be rescued. So it was not 100% dead at the beginning, but if you're not treated properly, it could be 100% dead, which is obviously not good for you. You have half of the brain dead, which is not good. So if you have a timely treatment or reperfusion, it may occur that the penumbra is 100% rescued. So you're left with this only very small infarct area. So now the goal of ischemic stroke neuroprotection is to really protect the penumbra, because the infarct zone, people usually believe it cannot be rescued. It is basically dead from the very beginning. So this concept, has evolved during the past 20 years. People are actively developing neuroprotectants to help maybe reduce cell death, reduce excitotoxicity, which is the excessive firing of the neurons, which leads to their final death. But lately, those attempts haven't been very successful. So people have switched gears and looking to the new concept of neurovascular unit, which is the integral part of this multicellular component that includes not only the neurons, but also the vascular side. For example, the endothelial cells, the pericytes, and also the inflammatory side, which include the two key players, microglia and astrocytes. So people start to believe that protecting the entire NVU would give you better results rather than protecting only the neurons. So with this, we also started a few research projects in this area by looking into the components in the NVU other than neurons to see if we can, perturb those components to help rescue the entire NVU, and in the end help rescue the neuronal behavior or the infarct, the penumbra, and the outcome. So we started from also the cell level. So we looked at this the endothelial cell side, the brain microvascular endothelial cell, BMECs. So we have this early effort of we formulated a systems biology model of the BMEC signaling that involves a number of the pathways that play a role in the signaling and the fate decision of BMECs. And then we, as the usual systems biology model would do, we use a number of experimental data, the time course, signaling, drug perturbation, dose response, phenotype to calibrate the model. And then we did a sensitivity analysis. We try to find new targets, and then we perturb the proposed targets in our virtual virtual BMEC cell, if you wish, to see how much perturbation would lead to how much phenotype reversion in an adverse event of the ischemic stroke. So basically, if you do experiments for ischemic stroke, we would start with in vitro level, where we culture the cells in a condition called OGDR, which is basically oxygen glucose deprivation and then reperfusion. So we withdraw the oxygen or glucose in this chamber for a while, usually for a few hours, and then we reperfuse this chamber with back with oxygen and glucose, because in the real ischemic stroke clinical setting, that's usually the reperfusion occurs, right? You have the recanalization, you remove the clot, and then suddenly your brain is full with blood again. But that's not really necessarily good. That could lead to secondary damage due to the reperfusion injury. So anyways, we simulated this virtual BMEC in this OGDR, experimental procedure computationally, and then we see that there is this node called the succinate, regulated by SDH, came up as one of the top hits from the computational analysis. So then we searched the literature. So it's also a new target for BMECs. Not much research on this in terms of the BMEC regulation, nor in the ischemic stroke. So we selected one of the inhibitors of this process, malonate, and we first did experiments in vitro setting using the exact same OGDR procedure. So we see that by adding malonate into this environment, we did reduce the inflammatory phenotypes, which are predicted by the model, including the IL-6 as one of the greatest markers. And also we restore the tight junction behaviors by looking at the Z01, which is the tight junction protein. And also for the oxidative stress side, adding this inhibitor did reduce the oxidative damage happening to the cell. And also we did some microscopy to further validate it. But more importantly, is this beneficial for real, in vivo setting and the endpoint? So we did, again, advance into the preclinical mouse setting by performing the TMCAO, which is the transient middle carotid artery occlusion model, mimicking the ischemic stroke, the acute ischemic stroke. So we, again, added this malonate, and there's vehicle, there's sham group. We, again, so micro dissect the endothelial component of the brain tissue. So basically the microvasculature, we observed the things changing in the microvasculature. The same thing so agrees with our previous computational prediction, as well as the in vitro experimental results. We see decrease in the inflammatory markers. We see the restoration of the tight junction proteins, and also the reduction in the oxidative stress, which is the nitrosidative stress. But in the end, we also have reduced brain damage due to the addition of this drug. So this is vehicle, this is sham. Sham, you can see 100%, good brain. Vehicle, no drug, but with TMCAO, you see a large white area, which is due to the TMCAO procedure that is called infarct, infarct with penumbra or non-rescuable penumbra. But with the addition of malonate, it really, recovers quite a lot, although not 100%, but it recovers quite a lot. So you see in this figure of infarct volume, which is the one of the key assessment for the ischemic stroke. We also see reduction in the neurological deficit score, which is meant to evaluate how good the mouse behaves in terms of functional motor activity. So the lower the score, the better the outcome. So it does rescue both the endpoint and also the neurological endpoint. So this is good. So this results in a publication. But now we are advancing it to the in vivo setting. So we have formulated this in vivo QSP model of mouse stroke. Basically, we even attempted it from two perspectives. One is from the acute side, one is from the chronic side, using two different animal models. One is TMCO, as I just introduced. The other one is called BCES, which is basically chronic ischemia in the mouse brain to mimic the chronic aspect of the stroke or cerebral small vessel disease. And we are also advancing into the human to try to make a translational prediction. So I'm just showing the animal part of this is just showing the animal part, the mouse QSP TMCO model. So again, we calibrate the model using tissue level observations for different cells, for different cytokines, different molecular markers. And then we can generate this in vivo prediction of the infarct area. Basically, you see a sudden rise in the beginning due to the TMCO occlusion procedure, and then after reperfusion, sometimes it decreases a little bit, sometimes it doesn't decrease, sometimes it even may increase. And then, as I introduced previously, similar to the PAD work that we've done, we generated this virtual mouse population that all went through TMCO. So basically, virtual TMCO mouse. And then we perturb different nodes in this system. But for this time, we try to see if we can do any combinations, because for ischemia, maybe combination could be the future path to go. So we did identify a few combinations that are predicted to enhance the infarct or the reduction of the infarct after reperfusion. So we selected the most prominent one and we did some experiments. So this combination is on top of a traditional, inhibitor of necrosis, which is the same NEC1S that we used before, but this is on top of that, we added something new. So basically, after you went through the similar experimental procedures, you see vehicle group, you have large wide area, NEC1S somewhat less, but for this combination, it again significantly reduces the infarct zone, as seen in this bar plot. Also, if you see in this actual brain image, you see this indent, deformation, which tells you that your brain is certainly not good. But for this combo, it more or less is very close to the original status, but not 100%, as I said. And we also domesticated several detailed markers. For example, the apoptosis and necroptosis markers, much less in the combination compared to the single agent, also to the control. And also for the inflammatory side, you see the pro-inflammatory, anti-inflammatory, they are more or less modulated or towards the more balanced environment. And also for the motor behavior tests, for example, the rotor rod, the combination leads to higher scoring compared to the single agent, also comparing to no drug, similar to control, I would say. So this is, I think, a very good sign for this model-informed approach that we have at least validated one pair of combination. We are actually doing the second pair at the moment. So this is manuscript is in preparation. So as I said before, we are also advancing this QSP model to the clinical stage. Of course, we need to make some modifications to the model structure, which I don't have time to go through today. But for the human side, we have much less data. Usually, you would only have some peripheral biomarkers. And we also collected some CSF biomarkers from the literature, from the experimentals, from the clinicians. And then you're left with only the endpoints, basically. Either it's the NIH-SS neurological scoring, MRS scoring, or the imaging, the results, for example, how much infarct volume is in each patient. So we, in our first attempt, we try to link this, the infarct volume, which can be measured radiologically in each patient, try to link this infarct volume measurement to his or her NIH-SS score. But unfortunately, we only have 2 points of NIH-SS score. One is during the admission stage, the other one is after surgery. So we, again, used, I think, 50-ish patient data, try to calibrate the model. We can calibrate them, of course, but the thing is that we're still expecting more and more data for us to establish this relationship, because at the human level, we only are scarce of data. So in order to have a good virtual population, we need to have more individual patient data. So this step is still ongoing, and maybe I have time to report to you in the future. So that's for the CNS cardiovascular side. But one thing that we also have a project on MDD, which is a depression, which are in progress, which are also equally hard to do, because for depression, the endpoint is not any biomarkers, it's still scoring or the intent to suicide, commit suicide, or something like that. But again, we're doing actively on that. For the cancer side, so many of you may know that previously when I was at Hopkins, our big group that led by Sasha Popol, we developed this huge model of QSP model for immuno-oncology that has also involved many talents. So let's just quickly go through it. This was a big model for the immune interplay with the cancer side that has advanced, if I remember correctly, to 600 parameters lately. It also involves a lot of people, so it is actually a collection of publications, 20-ish publications that lately Dr. Popol has included spatial omics, pathology, digital pathology into the model. So it's quite complex, but it's very seminal for this field. So recently, but my group, after I get back to China, is getting into this point mutations, which are in some ways simpler to do. For point mutations, there are very good publications early on by Genentech and Astellas, looking at the MAPK pathway, how we can use such types of models to advance clinical development of new drugs. So two projects that we are actively working on or partially completed is for MET and BRAF. So first I'll talk about MET project. So for MET, we're looking at MET overexpressed or MET mutated non-small cell lung cancer, because this is actually one big point mutation class in non-small cell lung cancer. The overexpression, the patients can count up to maybe 70% of our non-small cell lung patients. And a few of them, 3 to 4%, have this known exon 14 skipping. And for the MET pathway, people or the industry is actively developing new drugs, not only just the tyrosine kinase inhibitors or the kinase inhibitors, but also nowadays, bispecific antibodies like this one, or the ADCs, or even the bispecific ADCs. So I think this, at the beginning, we think it should be a very interesting project. So we formulated this model, already published last year. So formulated this model, primarily looking at the MET, but we also incorporated EGFR and some 2 other, mutation genes or receptors, ROS and ALK. And for the downstream part, we are primarily looking at the traditional MAPK or the PI3K-AKT that will ultimately lead to cell growth. And during this process, you have a lot of nodes that can be targeted by drugs, by different TKIs or kinase inhibitors. And even on the top, you can maybe include monoclonal antibodies, bispecific antibodies, or ADCs. So similar to traditional approaches, we employed a number of signaling data, drug perturbation signaling, dose response, cell viability, combination cell viability, and then to the animal level, you have in vivo tumor growth inhibition of CDX, of PDX, of monotherapy, of potentially combinations. And then we can formulate this virtual population at the clinical level. For example, we did this first attempt in this representative exon 14 skipping patients. So we can calibrate this virtual population using published data of 4 different MET inhibitors, the cametinib, tepotinib, crizotinib. So, and the other one we actually left for validation, but it turns out pretty good. And then we kind of, changed or tuned the parameter a little bit and formulated this MET amplified patients or MET overexpressed. They are more or less the same in our model. And we only have this one drug data for cametinib that have been tested in its MET amplified non-small cell lung cancer patients. But we can generate this population and simulate this response to other different MET kinase inhibitors, which haven't been published or tried or in clinical scenarios. So after we have this virtual population, so we can see that we predict the OR quite good. Not even with the individual patient level response, you can try to see if we can optimize the dose for each drug. But for here, we see that most of the drugs, at least most of them, their clinical dose is more or less around the plateau, but only for this, it has the potential to be reduced a little bit. That's one aspect of analysis. The other aspect, of course, is to look for optimal combinations. So we try to simulate the combination of any MET inhibitor, together with any other, PI3K or AKT or MEK or ERK inhibitors double combinations, even triple combinations, to see if it can enhance the overall response rates. So this is our analysis. So another attempt that we tried is to incorporate patient-derived organoids that we have 5 patients that are known to have MET amplification, and they were tested with a number of different chemo drugs or small molecule inhibitors, where you can see their experimentally measured in vitro PDO drug sensitivity results. And we can actually incorporate this into our model to project their clinical efficacy. But for the record, we haven't really tested those clinically, meaning that we really haven't clinically validated this model prediction, but this is just served to give you a sense of that. We can use the QSP model incorporating patient-derived organoid or organ-on-a-chip results to have clinical predictions. So again, we also tested new modalities, including the bispecific and ADC. So for bispecific, we included this marketed drug from Janssen that after we incorporated its in vivo data, clinical PK, we can somewhat reproduce its clinical PD using the exact same virtual population that we just developed. And similarly for this ADC, the MET ADC, we can also do the same. So I think this model also have a big potential for translationally simulating the new drug modalities, including bispecific ADCs, or even something in combination with a TKI, something like that. So another similar attempt was applied to BRAF. So BRAF, we are now looking at the class 1, 2, and 3 mutations in non-small cell lung cancer. So the early paper was only looking at class 1, the V600E mutation, but now we are looking at all 3 classes, because the 3 classes, they have slightly different origins. For example, the class 1 is just because of this overactivation of the gene. Class 2, it is actually have a impaired kinase function. And for class 3, it is even more impaired, and usually comes with RAS or some NF1 mutation that leads to constitutive activation of this pathway. So they have different causes anyways. So we formulated this model not only for EGF. So RAF is under, it can be under any receptors, but for non-small cell lung cancer, we specifically chose to cover EGF. And then we included both the FGF and IGF, which are also, reported to play some role in this resistance for EGFR kinase inhibitors and for BRAF inhibitors. So these 2 pathways could play a role in the secondary resistance type. So again, similar things that I won't go through the details. We use a number of data from the intracellular signaling, viability, preclinical tumor growth inhibition from all 3 different mutation classes, and then we can select some clinical data for validation, but this is actually very limited. The class 2 and class 3 mutations have been very less studied in the clinical setting. So the data here is also very scarce, but still we use all the data we can. We have access or we can pull out, and then we formulate this virtual population for class 1, class 2, and class 3 patients, and then we simulate the potential combinations for these different patient subgroups. So as the focus is for this more, less or understudied subgroups, we see that the combination of any MEK with a ERK could theoretically lead to enhanced response compared to using a traditional BRAF, some inhibitor like that. So this is very interesting because there's no publications or experimental validations for that. So we did do some experimental validations for this prediction. We actually use the actual PD, so patient-derived cells from actual class 2 and class 3 BRAF mutant non-small cell lung cancer patients, and we executed this mini PDX thing, which is similar to PDX but quicker, and so it's still engrafted in mouse, but we have results more quickly. So we tested this combination of trametinib plus this ERK inhibitor, low dose, high dose, and class 2, class 3. It all gives us a better response in this combination group compared to the single agent or compared to the control. So this is again, I think a good validation for our model prediction. So this part is also in revision, but we are advancing this part toward a more complex area. As the next step is to include the IO part. So for this IO part, we formulated a new IO module that are not the same QSP IO we had before, but a simpler, but I think more fit for purpose thing. We have the different T cells, the macrophages, NK cells, Tregs, because now we are looking into some new immuno checkpoints, the TIGIT, the PVRIG, and some associated ligands. And you see all of them over here. And then we can still do the same. We can formulate new or update our virtual populations using now immuno-oncology treated drug data. So there are a few trials that tested immuno-oncology agents, basically the mono PD-1, mono PDR1 in class 2, and class 3. So we use those data, but in my personal opinion, we probably are going to include more data because those data are really tested in very small numbers. But again, anyways, in our first attempt, we try to predict either double combination of an IO checkpoint inhibitor in combination with a small molecule kinase inhibitor. Would it result in enhanced response or similar response? If not, for a triple combination, would it result in enhanced response or similar response? So we did. There are of course different results with different classes, but we just used one of them. Double combination, and we did some in vitro co-culture for tumor PBMC using the class 2 and class 3 cell lines. And we did see that the combination could somewhat elevate or enhance the cell killing compared to some of the mono agents, and also significantly compared to the other one, but the extent is much less. So this work is still ongoing, yeah. So another thing that we have been working on is the HER2. So this is already published. We have this HER2 QSP model that included the different HER2, HER1, 2, 3, 4 pathway, the downstream activations, and more specifically looking at the ADCs and the ADC combination with TKI combination with chemo, things like that. So for those who are interested, you can refer to this publication. But in short, we included a number of cell signaling data similar to the previous sections, animal data, and then so for this scenario we predicted that a new combination could result in much less tumor growth or significantly enhanced tumor killing in this setting, and they require only very low dose for each of the drugs. So this is again validated in the model, and we then advanced to the clinical level by incorporating patient level, individual trajectory data, and population level OR data. And we can also include AE data, for example, cytopenias for A cell, for B cell, for C cells under this regimen, under that regimen. And then we can simulate potential combinations of a ADC plus a TKI to see if it can enhance any response or lead to, much worse blood cell adverse events. And if we lower the combination doses, would it give us similar results, or even have better, AE outcomes with similar efficacy results, to see if we can propose some new regimens for the clinical stage. And also for different lines of treatment. If we have TDXD resistant patients, then we treat them with TDM1, or we have TDMR resistant patients, then we treat them with TDXD. Would it give similar outcomes in the 2-year lifespan? Like similar ORs or similar at the 2-year endpoint, would they both have similar tumor regression? Of course, the results are not the same, but this part is still in prep. Also, we actually expanded that ADC model to a more comprehensive one. For that purpose, we can basically incorporate preclinical data, and then we can project the potential clinical efficacious dose for ADC, not the first in human dose, but the potential efficacious dose, and also the optimal efficacy dose for the ADC, which means that at that dose you're expected to have optimal ORs, but if the dose even goes higher, you wouldn't have increased ORs, but increased toxicity instead. So this is also in prep. So for the last part of my presentation, I'm going to go through some cell therapy and cell and vaccines. So for cells, we are primarily working on immune cells and stem cells. So for immune cells, of course, you are very familiar with CAR-T, and there are a number of publications in CAR-T for the modeling side. This is not an exhaustive list, so you can find several more in 2025. And also there are many good reviews for the how to use QSP models or mechanistic modeling or for cell therapy decision making from the very early on stage to the late stage. So I think I skipped this. The thing is that for simple PK/PD models, it can characterize cell behaviors only when you have a large quantity of data. But for mechanistic modeling, you can extrapolate from very small sample size and then make predictions for your next stages, for example, from phase 1 to phase 2 to phase 2 to phase 3, etc. So for this, we have produced this multi-skewed QSP model for CAR-T for solid tumors. This was partially published in the 2015 paper, where we incorporated the basic, CAR-T cell interactions, tumor cell leading to cytokine secretion, cell killing, and T cell proliferation, and then we can incorporate a number of drugs or not drugs, a number of CAR-T products targeting solid tumors, where we can obtain their preclinical data and clinical data from literature. And then we also have this in-house product of a Claudin 18.2 targeting CAR-T. We have their own preclinical data from in vitro to animal. So we can use this QSP model to incorporate all this data to train some relationship to help us predict the first human dose for CAR-T targeting solid tumors. And also this model, as shown in this publication, can help with dosing regimen design. For example, if we want to treat heavy or light patients, or should we use a kilo-based dosing or flat dosing? Would they result in similar efficacy? If not, how do we adjust the dose for heavy patients, things like that? And also, if we want to split dose, for example, should we use just 1 dose at day 1 or 3 dose at day 1, 4, 7, or 0, 1, 2, 3, equally split or step up manner? Would this result in similar efficacy and toxicity? So this QSP model can help to do things like that. And the things that are still in progress or haven't published is that so as you see here, we actually included a number of cell subtypes for this CAR-T product, not just for CD8, but also for CD4. So we can actually, again, fit those individual patient data because we have the individual patient data for all those different cell subtypes in blood after infusion in patients, and also together with our tumor regression data and a number of cytokine data. So after that, we can formulate this, large sample size virtual patients based on only like 20-ish or 10-ish patient samples. And then we can simulate potential dose response in a more precise manner. So that's one thing. The other thing is that we can test maybe different combinations of cell compositions. For example, should we have more naive cells or should we have more effector cells? How would that affect the clinical endpoints and efficacy outcomes in the clinical trial setting? So this is, I think, very interesting work. At least we showcase the potential utility of this in a very early on drug manufacturing setting, and because this happens usually at the preclinical stage. We are actually also using this methodology for another cell, or another type of immune cell called tumor-infiltrating lymphocyte TILs. So for TILs, it's different to CAR-T because they have no CAR, but they are more diverse. So you can target different numbers of different types of cancer clones. But they comes also with their own problems. That one thing is that their clinical dose is a very big range, usually maybe 10-fold up and down, or even 50-fold up and down. So that's one thing. The other thing is that usually you would infuse some cytokines to help boost the TIL proliferation in your body. Or now people are armoring TIL with some, IL-15 or things like that into the product. So for the early on, how would IL-2 infusion impact the TIL efficacy and toxicity? What is the optimal dosing regimen for IL-2? So it all comes with this TIL thing. So we are also collaborating with some clinicians and industry partners that they have TIL products in clinical testing early on. And we are also using this multicellular approach you see here, but now for TILs. So we included CD4 and CD8 subtypes, and also some specific TIL-related subtypes. CD39 negative, CD69 negative, and PD1 positive, and also the host TILs, because they are originally there, but we are just infusing more. And also we have IL-2 and its impact on cell proliferation. How would it lead to different cytokines from TILs, from IL-2, from chemokines, and also it has some blood cell issues. For this, I'm not going to show the full results, but still similar things. We simulate this individual patient level, individual cell marker, cytokine, chemokine, tumor lesion change. And then we can formulate virtual populations then to simulate first the different IL-2 regimens, like 1 dose or 2 dose or 6 dose, up to 6. And this dose per shot, that dose per shot. How would it impact the final outcomes? For example, efficacy, if we have ORs and PFS as our outcomes, and then toxicity, we would count on cytokine release and the blood cell. So this is also in progress. We are actively working on that. So another thing that has been published is this work on stem cells. So we had this, early on, we did this project, try to use QSP modeling to help design the optimal dosing regimen for mesenchymal stem cells for lupus nephritis. So a very complex autoimmune disease, but a very complex drug. So for this goal, we have completed this goal in 3 steps. First, we built this model for LN, so lupus nephritis themselves. So only the disease pathophysiology by including different cell subtypes, different cytokines, different efficacy markers. Try to mirror the model by using healthy patient data, healthy volunteer data, and also patient data. And then we use this PBPK-based approach to project the in vivo disposition of the MSC and mesenchymal stem cells in the patient level. So for this one, we have this PBPK model, and then we use mouse data, rat data, and then we actually have human data measured by radio labeling, very few patients that can serve to some extent as a validation. So for this part. So the third component of our model is to, so this is just showing the mouse data, rat data we used, not showing the human data here, but again, this too. So for the D and not showing in full here, but we have some literature imaging data, and we have our own imaging data for this MSC product in human level. The third component, as I said, is to simulate how do I put it? The SOC, standard of care treatments, because we're going to simulate a double-blind clinical trial. One group is using standard of care. Basically, you have MMF or prednisolone or cyclophosphamide, which are immunosuppressants. On the other hand, on the other arm, you have SOC plus MSC. So we will want to simulate these 2 different scenarios. So of course, we should first validate our model using clinical level data by using only the SOCs, because you would hardly find any MSC trial data from the literature. So first, the SOC data is all we have. We simulate our trial using different SOC regimens, and we see if our model can reproduce those clinical endpoints, including the renal biomarkers, including the peripheral biomarkers for not just one trial, not just one dosing regimen, for different trials, different dosing, 1 gram per day, 2 gram per day, 2.25 gram per day, 1.5, things like that. So we utilize a number of trials that are on SOCs and their biomarkers for our model calibration. And we also have this, IIT data, internal IIT data that we tested some MSCs, just monotherapy in patients, and some very short-term measurements on the endpoints. So it actually matches our model prediction quite well. Not for the record, but we again validated this model using our phase 1 data in lupus nephritis, but they're not supposed to be shown here. But anyways, we again, after single patient calibration, so we formulated this patient level of patient population by incorporating patient variability, and it turns out that this new patient population can reproduce the dynamic treatment response in terms of the duration or the response categories. For example, you will still have some complete renal response patients, partial renal response patients, and together we have this overall objective renal response that are different in different time points and also in different treatment regimens. So we validated the model using literature data. So the next step is to predict the optimal dosing regimen. So we used our MSC of interest, some parameters related to that. we simulate different dosing regimens. For example, 1 shot or 2 shots with 7-day intervals, 2 shots with 1 month interval, 2 shots with 3 month interval, 2 shots with 6 month interval, both at the low dose SOC combination, high dose SOC combination, not only for single patient, but also for population. The graphs are not shown here, but in the publications. But the result is that we conclude that 2 shots with a 3 month interval would give you best possible outcomes at this, endpoint you are interested in. So this new regimen was later adopted in a phase 2 design for a original MSC product in clinical development in China. So we again also try to, see if we can, MSC would bring any benefit, even in the resistant or highly resistant patients. So you see here, we created this highly resistant patients that are in this lighter blue line. You hardly have any effect under the SOC regimen. But when you combine those with MSCs, it sort of gives you some reduction in this proteinuria level. Not very good, but also it already gives you some patient benefits. So if you use this new metric called time of proteinuria under 2, that we can show that in this highly resistant patient population, the addition of MSC will still give you some benefit in patient response. That's one thing. So another thing is that we try to see if we can find some new biomarkers to help stratify patients so that the stratified patient subgroups would have a more improved renal response to the SOC plus MSC regimen compared to SOC alone. So this is our simulations of a pulled together population that only you see very minor improvement in ORRs of SOC plus MSC compared to the SOC alone. But after we do some virtual population analysis, by selecting those top ones compared to the bottom ones, we did find that if we can, use this thing as a new biomarker, for example, the high CXCL10 plus low UPCR or low proteinuria, then we would potentially elevate this OR difference to something like 20-ish. This, of course, is very good and very desirable. But so we are actively looking at how this can be implemented at the clinical level. First, we might need to do some retrospective analysis by using patient samples, but this is still ongoing. So similarly, we incorporated some new targets. For example, the B cell related targets like the BAFF/APRIL, BAFF receptor inhibitors, BAFF antibodies, BAFF/APRIL dual antibodies. So this is just showing a case study for BiMed-Math, which is a BAFF antibody. And then we plug in its affinity from in vitro, their PK, and we have a minimal PK for its extrapolation for renal concentrations. And then we can somewhat reproduce its clinical endpoints for the complete renal response rates over time. So we are also testing more and more components into this QSP framework. And this is for lupus nephritis, but we are also expanding it to another autoimmune disease called systemic sclerosis. And also we have the same MSC product being developed in this indication, and now it has completed phase 2. We are using this QSP model not just to explain its efficacy, but to explore if we can formulate some virtual patients or digital twins to mimic the patients, because this is a rare disease. So we don't have many patients, we don't have many dose groups. So if we can use QSP model to mimic those patients, we can somewhat create a larger population and simulate more what-if scenarios. For example, longer time spans, longer follow-up durations, and more dose combinations, things like that, to help with the evaluation of its clinical endpoint. So the last 5 minutes, I'm going to go through some examples on vaccines. So vaccines, as you know, for the prophylactic part, there are many good publications and many good ongoing work from the different US companies or European groups. And for that, we also established or formulated this project on COVID, using COVID as an example, but we are now looking into 3 major different categories of vaccines, including the mRNAs, the adenovirus, and also the whole killed. So this work is still not published yet, but we have this, a similar framework that are expanded from the very early on Chen's work for immunogenicity, but we expanded it. We included more details in the dendritic cell processing of the antigens. And then we can simulate the first biomarker response in the different modalities, the mRNAs, the adenoviruses, as well as the whole killed, and also not just for cell markers, also for antibody levels, also for the population level antibody levels. And because of the fact that we have different modalities, we can simulate the different combinations of vaccine modalities. For example, whole killed plus adenovirus plus mRNA, whole killed plus mRNA, things like that. 2 shots, 3 shots. So this is just showcasing classical analysis you would have for a model agent. If you should have an 8-week interval, or 4-week interval, or 12-week interval for your vaccination shots, how would it result in your differential response of population level protection? And if you can have 2 shots or 2 combinations, combination of 2 different agents, modalities, how would it impact your overall protection? So this is being simulated in our model right now. Another side is for the therapeutic
Chen?
part.
Hi,
Yeah.
Chen, can I just interrupt you? Yeah, is the time is
It's closing
almost
soon.
ready.
Yeah, like maybe
So-
2 minutes at most.
Okay,
So
thank you.
going through very quickly, so we have a project on HPV or for HISO, so we can simulate some therapeutic response for mRNA vaccine. So basically LNP encapsulated mRNA vaccine to treat these patients, and we can, by leveraging early phase data, we can construct, similar virtual populations to simulate more clinical scenarios for translational research. So this is the end. I'll skip summary, and I would like to thank all my previous collaborators in the US, in China, my own group, and also our industry collaborators. Thanks again for inviting me to have this chance presenting our work. Thank you.
Thank you very much. Really nice talk, especially in the beginning is the history of the QSP model, and then how did the industry people meet the academia and pursue the development of this QSP. Was really amazing talk. And then your recent work was great.
Thank you.
Yeah.
Thank you.
Yeah, and then actually we don't have such a much time for the questions, so we want to quickly to the advisors' questions. So let me start with CJ.
Hi, thank you very much. And thank you, Chen, for that really excellent presentation.
Thank you.
Really
Thank you,
going
CJ.
from insights to where QSP can have the most impact to many different examples. And I think, really taking the audience through the preclinical work to, the translation to person human to then to clinical outcomes. That's really a huge leap that I don't think that many people appreciate how difficult that is to do. And so I really commend you on that. One thing I wanted to ask you about as I was looking at all of your different examples was going back to the ischemic stroke, then to oncology, where we're looking at genetic mutations. And correct me if I'm wrong, but I believe that ischemic stroke is actually pretty high in Asian populations. And when you think about mechanistic modeling and where you're going from the preclinical mechanisms to clinical, have you thought about going back to the genetics for that?
Thank you, CJ, for this question. We haven't done that yet because, the mechanistic origin for ischemic stroke is already very complex. But I think this is a valid point to be maybe added in the future for some, genetic things. Yeah.
Yeah, sorry. That was a very difficult question, but it struck me as I was going through all of your examples. So thank you so much.
You're right. Yeah, thank you.
Okay, then I would like to go to Professor Kenji Kabashima. Kenji Kabashima, Professor? Yeah.
Thank you for nice presentation. And I just wonder whether how do you combine with your model to clinical questions? So some quick clinical questions might be difficult, may not fit well with your model, but so how do you decide to your research topic?
Yeah, I think that's a question that we should pretty much solve when we start a project. Usually we start a QSP project when there is either a product or a product in active clinical testing early on. So the question would become dosing regimens. That's a very usual thing for, especially for cell therapies that I've showed, or the mRNA therapeutic vaccines, things like that. The other one is to look at those understudy areas, like the class 2, class 3 BRAF. Those are really understudied in the clinic. But people are looking at new combinations. It's just that our QSP model, as I believe, would offer a new route to provide some new insights that for either preclinical translational research or future, IIT studies in a clinic, because those drugs are already on the market. That people can initiate IITs if they find this results interesting. So I don't have a universal answer for your question, but the good thing is that I keep a lot of communication with the clinician side and the industry side. So we see questions quite a lot. So at least QSP model can solve part of them to help speed up the product development. Yeah.
Thank you.
Okay, so if there are any question from general audience, I would like to take one question. If not, okay, we'll go to the next advisor. It's Professor Motoko Yanagida.
Okay, thank you very much for your great talk. I'm very, very fascinated. And I'm a nephrologist, and I'm really interested in the side effect of the molecular drugs. So if you happen to have a kind of whole body modeling, is it possible to predict the adverse event of the new molecular targeted drugs?
Yeah, I think that's a question that we hope to answer. But the thing for QSP, or the shortages for QSP, is that usually we should have some hypothesis for that potential AE in mind related to that new molecular drug. Then we can try to simulate its profile, the extent, or different dose response in the clinic. So, or depending on what the potential AE is. Yeah.
I see. Thank you.
Okay, there's no question from general audience. You can also directly remain the message in your questions. Okay, then we are going to the Dr. Kawai.
Yes, thank you so much for your great presentation. I have a similar question to CJ. So, for ischemic stroke model. So my question is, how did you adjust your model to human being? Because you have limited human being data. So I'm interested in how to adjust your model to human beings.
So as you pointed out, so human data are very limited. So we basically used a very similar model structure compared to our mouse side, but we changed, I think, maybe 5 different aspects. So one is that the compartment, I think we included additional compartments in the human version compared to the mouse version. And the other thing is the time of ischemia. So in human, it's very different to mouse TMCO. So we changed that. And then I think we tried, or at least we're trying to incorporate the different, so where the occlusion happens, here or there, or there are different categories. So that's one thing. But again, as you pointed out, there are very scarce data for human stroke compared to mouse models. And we are really using all we can. And we are, as I showed in my slides, incorporating more and more individual patient level data to hope that we can establish some connection between 2 biomarkers, 2, 3 biomarkers, or 4 biomarkers together with your NIHSS score, so we can predict the behavioral outcomes. So yeah.
Okay, thank you very much.
Okay, so I would like to take a question from general audience. Okay, so Jinghao, if you have a question, you can just mute off and then can give a directly your question.
Oh yeah. Thank you for your talk. Yeah, similar question to Yanagida Sensei just asked. So is there any any way in the framework to discover like potential important pathway that's not pre-identified network? That's
This question. Personally, in my attempts or previous experience, we haven't been successful in doing this because that's beyond our thinking. But now I think, so for example, in our ischemic stroke studies, we usually start with some omics analysis, try to figure out some potential pathways. But for example, there are maybe 10 pathways coming out or 20 pathways coming out, but not all of them are included in our model in the first version. But afterwards, either it's because new information coming out or new publications coming out, we might, add one or two or a few more. But that's the only way that I can think of that may be helpful in, getting some data from outside your prior hypothesis from omics. Yeah.
I see. Because I have seen that some like applied mathematicians, they so instead of like go through literature research to build up the interaction network, but like you only identify like the how do I put this? So they basically traverse like the entire network to like the thousands of models and to see what like to test them. But that may be computationally expensive.
Right,
So,
right. For QSP models, I personally don't believe that we can traverse the 1,000 different versions for the model.
Right.
But I think starting from omics is good. You can at least have biologically supported information on the pathways you should include in your model, not just some random things that you add or throw in. Right. Yeah.
Yeah. Thank you, Bertha. That's impressive.
Okay, thank you very much. I think it's time to close our MMP seminar. And even in this time, I think more than 15 the participants from more than 15 countries. And then in the beginning, almost did 100, about 100 participants attended. And then still there remained more than 70 participants. So it was really great the seminar, and I really appreciated Professor Chen Zhao, his amazing work, and then the contribution to this medical and then pharmacology. And then I also would like to I really would like to thank all the advisors. They are very, very busy person, but attended all this full time. Thank you very much. And I also thank all the audience to attending this MMP seminar. And the next MMP seminar will be held in September. And please looking forward to who is the speaker. It's a secret. Okay, thank you, everybody. And then Dr. So is thank you.
Thank you. Yeah, looking forward to your next seminar and future communications. Thank you all for attending. Yeah, bye-bye.
Bye-bye.
Bye.